this lecture, we are going to examine structure of terpenes and terpenoids. Terpenes are a class of lipids that are formally derived from isoprene. Isoprene is an alkene, 2-methyl-1,3-butadiene. Formally derived means that terpenes are not really actually biosynthesized from isoprene. Instead, we can recognize isoprene structural element as constituent of terpenes. So a terpene may contain only one isoprene structural element, only these 5 carbon atoms of isoprene, or it may contain 2 isoprene structural units put together, or 3, and so on. If it contains several isoprene units, then that's an oligomer of isoprene. If it contains large number, then it's a polymer of isoprene. In living organisms, terpenes are actually synthesized for isopentyl pyrophosphate. You can see that isopentyl pyrophosphate is a derivative of isoprene. It has the same skeleton, but also it has pyrophosphate leaving group. That's very good leaving group. That's what makes it very reactive and suitable as starting material for biosynthesis of terpenes. Terpenes are classified according to the number of isoprene units that they contain or that they are composed of. So terpenes that are composed or have only one single isoprene unit are called hemiterpenes. And that's in part based on the old nomenclature, because originally when terpenes were identified and recognized as a separate class of compounds, terpenes that were isolated had 10 carbon atoms, and there were none with fewer than 10. So it was thought that maybe 10 carbon atoms was a basic unit. And so those were named monoterpenes. And now when it turned out that actually it's 5 carbon atom basic unit isoprene, then they had to be named somehow, and so they have been named hemiterpenes, which is Greek, it means half terpenes. So there are relatively few of those in nature, but hemiterpenes have 5 carbon atoms or 1 isoprene unit. Then monoterpenes, 2 isoprene units or 10 carbon atoms. Monoterpenes are very common in nature, and you will see some examples soon. Sesquiterpenes, sesqui is Greek for 1.5, that means 1.5 unit, are actually composed of 3 isoprene units or 15 carbon atoms. Then diterpenes have 4 isoprene units or 20 carbon atoms. Next one, make sure that you are careful about pronunciation, are called sesterterpenes. They are composed of 5 isoprene units or 25 carbon atoms. Sester means 2.5. Triterpenes, 6 isoprene units or 30 carbon atoms. And finally, we have one somewhat different class of terpenes, and those are tetraterpenes. Tetraterpenes have 8 isoprene units and 40 carbon atoms. You will notice that there is no class of compounds that has 7 isoprene units or 35 carbon atoms. That's because biosynthesis, by adding sequentially 5 carbon isoprene units one to another to form larger and larger terpene, stops at triterpenes at 30 carbon atoms or 6 units. Tetraterpenes are formed by binding 2 diterpenes together. So tetraterpenes are actually dimers of diterpenes. Here are some examples of commonly occurring terpenes. So here are some monoterpenes, like myrcene, which is an open chain terpene. Then limonene, component of lime fruit. You can see it has one 6-membered ring. Then alpha and beta pinenes, rather complex structures of complex bicyclic structures. They're components of fragrant components of pine oil. And then geraniol, very pleasant smelling compound that is a major fragrance component of geranium oil. So from these examples, you can see that terpenes are actually secondary metabolites, and they have diverse functions in living organisms. They are often main components of essential or fragrance oils. Some play a role as protective chemicals, and so they may be antifeedants or insecticides, maybe bactericides or fungicides. And other terpenes may play a role in communication as either pheromones or hormones. Pheromones are used in communication between different individuals of same species, while hormones within the same organism, between different organs or different tissues. Medicinal herbs are usually rich in terpenes and terpenoids. And terpenes are considered to be in part responsible for their pharmacological activity. Terpenes are derivatives of isoprene. Terpenoids are modified terpenes. So they have terpene as starting material, and then biosynthetically they have been modified, so their structure is a little bit different. So now we can see some examples of sesquiterpenes, farnesol, another important fragrance compound, and then partenolide. I believe that one is a protective chemical. And a couple of examples of diterpenes, again protective chemicals, taxadiene and abietic acid. Tetraterpenes are also known as carotenoids. Carotenoids play a role in photosynthesis and vitamin, so like beta carotene, and you can see that tetraterpene is composed of 8 isoprene units. You may be able to identify them here. Beta carotene is actually a precursor of vitamin A, but you can see that actually if you put together two vitamin A structures, kind of head to head, where OH groups are, you end up with beta carotene. So you can see sort of in reverse fashion how tetraterpenes are formed by attaching together, fusing together two diterpenes, because vitamin A is a diterpene. It has 4 isoprene units, while beta carotene is tetraterpene, 8 isoprene units. Steroids are a large class of lipids that have common structure. They contain cyclopentanoperhydrophenanthrene skeleton. They are terpenoids, which means that living organisms synthesize them from terpenes. And in this case, starting material is terpene squalene. Steroids have a number of different subclasses. There are literally hundreds of different naturally occurring steroids, and they have considerable biological and medical importance. So steroids could be, some class of steroids are sex hormones, adrenocortical hormones, bile acids, cardiac glycosides, saponins, steroid alkaloids. So their structure is, to explain their structure, we should start with aromatic compound phenanthrene. So those are 3 benzene rings that are fused in what we call angular fashion. Now, if phenanthrene is completely hydrogenated, so that it's completely saturated compound, then that compound is called perhydrophenanthrene. Perhydro means that it has been completely hydrogenated. It has as many hydrogens as basically it can take in the course of hydrogenation. And then, of course, you know what cyclopentane is. So when perhydrophenanthrene and cyclopentane structures are fused, we end up with cyclopentanoperhydrophenanthrene structure. This structure contains 4 rings labeled like this, A, B, C, D, so that's steroid designation, and numbering is as shown here. It starts with ring A in clockwise fashion, ring and A are numbered, then counterclockwise ring C, and then again actually rings A and B, sorry, I made a mistake. Rings A and B are numbered in counterclockwise fashion, 1 through 10. Then ring C is numbered clockwise, starting at 11. And finally, ring D started at 15 counterclockwise. And then after that, any substituents on cyclopentanoperhydrophenanthrene skeleton get higher numbers. In animals, the most important and the most abundant steroid is cholesterol. And here is numbering of substituents also on cholesterol. So structure of cholesterol and numbering of substituents on cholesterol. In a membrane, cholesterol molecule is oriented parallel to the fatty acids of the phospholipids, and hydroxyl group interacts with phospholipid head, with polar head. Cholesterol is not present in prokaryotes, and some of the intracellular membranes of eukaryotes. But it gives some rigidity to the cell membrane when it is present. And cholesterol constitutes about 25% of the membrane lipids of some nerve cells. Cholesterol is biosynthesized starting with triterpene squalene. This is very abbreviated. Biosynthesis, so squalene in an enzymatic reaction is epoxidized, and then this reaction is like a zipper. As epoxide opens in a proton-catalyzed reaction, or proton-induced reaction, carbocation forms, which attracts pi electron pair from the neighboring double bond, closes six-membered ring, forms a new carbocation, which then attracts next electron pair from next double bond, and so on and on. And basically, like zipper, these double bonds form single bonds. Pi bonds are converted into single bonds, and all these rings, all this four-ring system forms very quickly. Eventually, the resulting carbocation in several steps is converted into cholesterol, and in the process, several rearrangements and losses of methyl groups occur. So at the end, what was starting terpene skeleton has been considerably changed. And so for that reason, cholesterol is not terpene, it's a terpenoid. You can see that its structure resembles terpene, but it's not exactly terpene. You cannot identify individual isoprene units, and that's because of the biosynthesis, because of the rearrangements and loss of methyl groups that occurred during the biosynthesis. So this completes our brief review of terpenes and terpenoids.